Hello, let's discuss on the topic micropropagation. Micropropagation is a rapid vegetative method of multiplication of plant materials from agriculture, horticulture and forestry purposes. Micropropagation is a highly productive method and it occurs in a fast process. Micropropagation is achieved by following two methods. The first method is by multiple shootlet production. Micropropagation result in development of shoot tips, and these shoot tips are used for tissue culture and raising mini plants. So the shoot tips in the culture medium produces multiple buds, and each of these buds later on develop into shoots. So by introducing a root developing or root forming hormones, these shoots can be induced to develop roots. So once root is developed, this plant body, these shoots can be transferred into other medium or into the soil, where these roots will absorb minerals and help in the growth of the root to become a fully grown plant body. Second is by somatic embryogenesis. Somatic embryogenesis refers to development of embryo from the somatic cell. Somatic cell here refers to the body cell. We usually during fertilization the zygote arise by fusion of male and female gametes, and this zygote give rise to embryo. But in this somatic embryogenesis, the embryo develop from the somatic cell, not from the gametes. So the embryos developed from a single somatic cell by tissue culture is known as somatic embryos or embryoids. Now let's discuss on interculture and formation of androgenic haploids. In interculture, androgenic haploid embryos, that is male, androgenic here refers to male. So male haploid embryos or plantlets are obtained from the microspore or pollen. The development of the pollen into the haploid sporophyte, that is embryo, in the culture medium is known as male parthenogenesis or androgenesis. If the pollen grain is treated before culturing with chemicals like colchicine, this pollen grain can develop into homozygous diploid embryos. Now, what is the significance of haploid plants is that these haploid plants can undergo mutation, and this can express themselves easily, and this expression can be easily detected because the haploids have only one set of genes. Second is that the homozygous diploids can also be obtained in a single generation by treating the haploids with chemicals in the culture medium like the colchicine. Now let's discuss on embryo culture. Embryo culture is the culturing of young embryos that is excised from the developing seeds and then culturing into the culturing in vitro, that means inside the laboratories on the nutrient medium. This embryo culture is utilized in some crops for improvement program to obtain the interspecific hybrids. Inter means different, so interspecific hybrids means hybrid that is developed between two different species. So this embryo culture is used to develop interspecific hybrids and also help to overcome dormancy and propagation of plants like orchids. Now in the interspecific or intergenic hybrids are usually sterile. Sterile means they lose the ability to reproduce. So they are usually sterile because the embryo mortality in this type of hybrids are very high and the seeds collapses easily. So in such cases, the young hybrid embryos can be excised from the ovary and then cultured in vitro. So this removal or excision of the young hybrid embryo from the ovary and culturing in vitro is referred to as embryo rescue. Now let's discuss on application of embryo culture. In some interspecific crosses, the endosperm of the developing hybrid seeds degenerate very early. So once the endosperm degenerate means the young embryos were unable to derive its nourishment, because this endosperm provide nourishment to the developing embryo. So once this embryo degenerate, once this endosperm degenerate, the young embryo become very difficult, or in many cases the embryo also degenerate because nourishment is not available. So in such cases, the embryo culture technique provides a hybrid seedling. That means it provides alternate method for developing embryo again. In some other plant species, the seeds remain dormant due to presence of certain inhibitors that prevent the growth or development of the seeds. So in such cases, the embryo culture also help in overcoming the sterility. Third is that in seeds of some plants like orchids lack stored food, that is they do not contain endosperm at all. So the embryo culture in these cases provides an alternative methods for development of seeds and seedlings. Now let's discuss on the protoplast culture and somatic hybridization. What is protoplast or protoplasm? Protoplast or protoplasm is the living content of the cell. The cytoplasm together with the cell organelles that form the living content of the cell. So protoplast is this living content of the cell. Now protoplasts are usually obtained by dissolving the cell wall using enzymes like cellulase and pectinase. So once the cell wall is being dissolved by this cellulase and pectinase, the inner layer, the remaining portion, is the protoplast. The protoplast can also be isolated from leaves, callus culture or suspension cultures, and from pollen grain as well. Now let's discuss on the protoplast culture uses or the purpose of protoplast culture. The first one is somatic cell hybridizations. A hybrid produced by the fusion of somatic cells of two varieties or species is called as somatic hybrids. Now the process of producing this somatic hybrid is called as the somatic hybridizations, and in plants this somatic hybridization is achieved by protoplast fusion. So somatic hybrids are usually produced by the fusion of cell of two different species. So somatic hybrids refers to the hybrids of two protoplasm. Now this process of production of somatic hybrid, that means the fusion of two different protoplasts, is referred to as somatic hybridizations. The protoplasts of different species can be fused to form different hybrids. Example, in 1978 the protoplasts of tomato and potato were fused, and the resulting hybrids is called as pomato. So pomato is the somatic hybrids that result by the fusion of the protoplast of the tomato and the potato. Now the other example is the protoplast fusion of the brinjal and tomato has also given rise to a new hybrid called as the brorato. So brorato is also a somatic hybrids which result from the fusion of the protoplast of the brinjal and the tomato cells. So these protoplast fusion is of high scientific interest and agronomic importance for raising hybrids between distantly related organisms. Now let's discuss on the application of somatic cell hybridizations. Somatic cell hybridization help in the production of hybrids between species that cannot be produced by sexual hybridization. Second is that gene transfer and the formation of transgenic plants can also be induced by this somatic hybridization. So transgenic plant development become very easy through the help of these somatic hybridizations, and even the transfer of cytoplasm also become very easy. That means once the two protoplasts are fused, there is obvious transfer of cytoplasm between two different cells. So transfer of cytoplasm between two different cells also become very easy. So these are some of the applications and advantages of somatic cell hybridizations.